In this video, we're going to talk about how Mendel principles relate to meiosis. And Mendel has two basic principles, the principle of segregation and the principle of independent assortment. So let's look at the first one, principle of segregation. This one states that for the two copies of each gene that a parent has, the offspring will only inherit one of those copies. So this means that those two copies will segregate and only one will be passed on to the offspring. Now, which one of those passes to the offspring will be decided at random. And this comes from the fact that most species are diploid meaning that they have two copies of all their genes, of all their genetic material, one copy that we inherit from the mom and one copy that we inherit from the dad. So in our genes, we have a set of genes that we inherit from our mom, and we have the same set of genes but with information that we inherited from our dad. That is what we call homologous chromosomes. We got 23 chromosomes from our mom, and we have the same 23 chromosomes, but now the information will be what comes from our dad. So during meiosis, what it does is it takes a diploid cell, which is a cell that has two sets of chromosomes. Like in the example here, we have a cell that has two big chromosomes, one red, one blue, one came from one parent, the other for the other parent, and two small chromosomes, again, one from one parent, the other for the other parent. So this cell will have two sets of chromosomes, four chromosomes in total. The two big ones will be homologous pairs, and the two small ones will be homologous pairs. So those two will have the same information that come in or code from the same genes, but will have different alleles. The first thing that needs to happen before meiosis is the chromosomes need to duplicate, so you need to make an identical copy of the information, and that copy will remain attached to each chromosome. So this chromosome duplicated and now has two chromatids, which each chromatid will have identical information. Now in this case, not only they have the same genes, but they have the same alleles because they resulted from one was the template for the other. And the same will happen for all of the chromosomes. So we still have the same four chromosomes. They're still called chromosomes as long as the chromatids are attached. But now each chromosome is composed of two sister chromatids, while here each chromosome had a single chromatid. The thing that happens during meiosis 1, here is where the cell goes from diploid into haploid. So this cell here had two sets of chromosomes. You see 4 chromosomes in total. After meiosis 1, the resulting cell now has only one set of chromosomes. So now we only have one chromosome of the big ones and one chromosome of the small ones. And the other cell has one chromosome of the big ones and one of the small ones. The chromosomes still have their sister chromatids, although now the chromatids have been recombined, so one chromatid might have a piece of the homologous chromosome and so on. We're going to go more into that. At this point, this cell is already considered haploid because it only has one set of chromosomes. So we already went from 4 chromosomes here to 2 chromosomes here. The next thing that happens in meiosis 2 is now we're going to split the sister chromatids. And from this, we result with 4 cells that are haploid cells. So all we did here was really split the sister chromatids from meiosis 2. So meiosis 1 reduces the number of chromosomes, and meiosis 2 splits the sister chromatids, the recombinant chromatids. So again, the importance of this part is because it takes a diploid cell into a haploid cell. If it weren't for that, once fertilization happens, we would duplicate the number of chromosomes. For example, in humans, if we have 23 pairs of chromosomes, that means we have a total of 46. Meiosis divides those chromosomes into 23, so only 23 go into the offspring. For example, here, this is a cell with 2 pairs of chromosomes, but we can imagine if it's a human cell, it will have 23 pairs, so a total of 46. And after meiosis, you will end up, for instance, in a mother with an egg that will be haploid. So we'll have 23 chromosomes with no pair. Then when fertilization happens, the sperm will contribute its 23 chromosomes and the egg will contribute its 23 other chromosomes. The nuclei of each of those cells will combine, and this is what we call karyogamy. Karyogamy is when the nuclei, the sperm and egg combine and form a diploid cell. So meiosis goes from diploid into haploid by reducing the number of chromosomes in half. Fertilization takes haploid cells from the father and haploid cells from the mom and combines them and returns them into diploid. So now we have, again, 2 sets of chromosomes, and this is a diploid cell. And this cell is the zygote, which is the first cell that will give rise to the embryo. So that was for the principle of segregation. So we go from having 2 sets of genetic information to just having 1 set, and that will be called haploid cell. And this allows fertilization, and fertilization will restore the diploid stage. The second principle from Mendel is this principle of independent assortment. Now this principle has to do with when we're looking at more than 1 gene at the same time. So for example, if we're looking at the gene for a round seed, whether the seed is round or wrinkled, and we're looking at a different gene that codes for the seed color, whether the seed is yellow or whether the seed is green. The principle of segregation says that a round seed, for example, is just as likely to be yellow or green, so that being round doesn't determine all the other traits, that the traits assort independently. So having 1 trait doesn't mean that you need to have other traits. So being round doesn't means that you can either be green or yellow. If you're wrinkled, you're also just as likely to be green or yellow. For example, if you produce yellow seeds, the plant can have either purple or white flowers. Having yellow seeds does not determine, the seed color does not determine the color of the flowers. Those are different genes that would assort independently. So let's look how this principle relates to meiosis. And this, one of the requirements of the principle of independent assortment, the easiest way to assure that it happens is when the genes are located on different chromosomes. And this is because different chromosomes assort independently. So let's look at this study. Here we have a seed that is homozygous for seed color, is for yellow, and for the shape is homozygous for round. If we can zoom in into the chromosomes of this plant, the large chromosome here, this blue stripe, it signifies the gene for round, and it has 2 sets of chromosomes, and since it is homozygous, each of those has the allele for roundness. And the small chromosome codes for seed color, and since it is homozygous for yellow, both of those also have the gene for yellow. On the other side, we have a homozygous seed, but this is for wrinkle. It's a wrinkled seed, you can see the shape. And the color is green, it's homozygous for green color. So the big chromosome that coded for seed shape now has the wrinkled gene, which is this pink one. And since it's homozygous, both chromosomes have that same allele. And the small chromosome that codes for color now has the green allele, and this is a homozygous for green allele. So this doesn't mean that, this is just for the sake of the example, you can have a heterozygous, and we're going to see that next. Since both plants are homozygous, they only produce 1 type of gamete. So all their gametes after meiosis will have 1 chromosome. Since both of their chromosomes have the same round allele, this one only produces gametes with round alleles. And since it is homozygous for color, it only produces gametes with yellow allele. This one, same thing, since it's homozygous, all its gametes are going to have the wrinkle allele, because that is the only allele that this plant has. Both copies are wrinkled, and it will have the green allele for color because it is homozygous for color. Now let's say that these 2 plants fertilize, they cross-fertilize. So now from those haploid cells in meiosis, we have the baby, the resulting offspring now has 1 allele from each parent. So from this parent, it inherited the round allele and the yellow allele, and from the other parent it inherits the wrinkle allele and the green allele. So now the offspring is heterozygous for both, for shape of the seed and color of the seed. And this is where it becomes interesting. So now this, if the principle of independent assortment is true, that means that this plant here will be just as likely to produce gametes that have round and yellow color, round and green color, wrinkle with yellow color, and wrinkle with green color. That is what the independent assortment means. If independent assortment did not exist, then the gametes that this plant will produce will look just as the gametes that his or her parents produce. But that is not the case. So with independent assortment, the gametes that this plant will produce can have any combination between round, wrinkle, and yellow and green. So some of them would inherit the color chromosome from, the shape chromosome from one parent and the color chromosome from that same parent. So this would be round and yellow. But it can also have the shape chromosome from one parent and the color chromosome from another parent. So it can have round and green, which is a new combination that we didn't have in the parent generation. Or it can have the opposite. So now I have the wrinkle from one parent and the yellow from the other parent. So this is again a new combination of chromosomes that did not exist in the parents. And this will be like this original parent. You have the wrinkle chromosome and the green chromosome. So these ones here are what we would call recombinants. These are new combinations that are now taking one chromosome from one parent and the other chromosome comes from the other parent. And this principle of independent assortment results from, in part, the random orientation of the homologous pairs during metaphase 1. So in meiosis 1 and metaphase 1, the homologous chromosomes come together and face each other in what is called the metaphase plate, so that they align at the center of the cell. And how they align? So here is the big chromosome from one parent, the big chromosome of the other parent, small chromosome from one parent, small chromosome from the other parent, medium chromosome from one parent, medium chromosome from the other parent, for example. And they can assort like this, like all the paternal ones on one side, all the maternal ones by one side. This is one of the possibilities. But they can also assort in all sorts of combinations. So here 2 will be from the father and 1 will be from the mom facing this direction, or the opposite way, or any possible combination. So think of how many chromosomes we have. We have 23 pairs. We have 23 to the second power possibilities. 2 to the 23rd power possibilities of chromosome combinations. So this is a ginormous number of possibilities. It means that once this split, this cell will have one chromosome that comes from the mom, and the other 2 will come from the dad. So this is where you're mixing the chromosomes to pass to your offspring. Another source of independent assortment happens during crossing over. So in crossing over, the homologous pairs switch pieces of information. So the chromatids now come together, and a piece of the chromatid from the mom's chromosome will swap with a piece of chromatid from the dad's chromosome, and now we'll have what we call a recombinant chromosome. So now this chromosome is mostly paternal, but it has a piece of the maternal one. This one is mostly maternal, but it has a piece of the paternal one. So this means that the genes in this chromosome do not come just from one parent. Now we have a mix between maternal genes and paternal genes, which is what the principle of independent assortment states. So meiosis contributes to independent assortment. In meiosis 1, when we have in prophase 1, the homologous chromosomes coming together and doing crossing over, swapping pieces of information. And then metaphase 1, where the homologous face each other, and you can have different orientations of the homologous chromosomes with maternal and paternal facing different directions. so that at the end of meiosis 1, you already have cells that have one chromosome from one parent and other chromosomes from the other parent, so they already mixed. The next mixing happens during meiosis 2, now where the sister chromatids that are now recombined, so if you remember they already swapped pieces of information, they will also face random orientation on the metaphase plate. So this sister chromatid could be the one that is only maternal facing this direction and the one that is recombinant facing the other direction, or it could flip so that the recombinant one could be facing that direction and the unrecombined one could be facing the other direction. And this will create again all sorts of possibilities for gene combinations or allele combinations. So that at the end you end up with 4 daughter cells, and they all have very different genetic information from each other. So this one has one completely paternal chromosome with a small one that is mostly maternal but a piece of paternal. This one has one that is small, it's only maternal, but the larger one has mostly paternal and a piece of maternal, and so on. So you have all different combinations. Again, if you look at how many genes we have, the number of possible mixes between maternal and paternal genes are really, really big. So just to summarize, meiosis contributes to genetic variation by reducing the number of chromosomes that allow karyogamy. So again, mixing, we go from diploid into haploid by meiosis, and karyogamy, which happens during fertilization, merges the nuclei of a sperm and an egg cell to go back to diploid. This is the related to the principle of segregation. Meiosis also recombines the homologous chromosomes by combining information between the sister chromatids and crossing over. It also randomizes the orientation of the homologous chromosomes during metaphase 1, so that we don't inherit only maternal chromosomes or only paternal, our offspring doesn't inherit just the chromosomes from our mom or just the chromosomes from our dad. The offspring can inherit a mix of chromosomes from our mom and our dad, so that the offspring has the random combination of its grandparents' chromosomes. And the last one is by the random orientation of the recombinant chromatids. Now it's important that they're recombinant because now they're different from each other, and how they face on the metaphase plate during meiosis 2, the sister chromatids plate during meiosis 2, also helps create variation in the possible combinations of alleles. So the first of those facts is responsible for the principle of segregation, and the last 3 are the ways in which we can have the principle of independent assortment. So this is how Mendel principles relate to meiosis, how they're the result of the events that happen during meiosis.